What I'd like to do is give a undergraduate view of complexity in the brain, and then talk about some of the ways we here at the Institute really think about dealing with that complexity. So a few simple statistics. A human being has 10 to the 14th cells. A brain has maybe 10 to the 12th neurons, 10 to the 11th to 10 to the 12th. And a human brain has about 10 to the 15th connections. So it is an unbelievably complicated organ. But it's also an organ that interacts with its environment in many different ways. And those interactions can profoundly change the nature of the connections that occur, probably the continued maintenance of many different types of neurons. And one of the major questions is, how are we ever going to be able to understand the emergent properties of the brain in terms of its fundamental components, molecules, cells, connections, and in terms of its environment and what the environment does to that network? And of course, the emergent properties of the brain range from consciousness, which in my view is the most challenging and difficult topic, through memory and learning and language and many other kinds of things. So how do we go from cells and connections to a fundamental understanding about properties of the brain? And the window through which we can actually study the human brain is really limited in interesting ways. It's been largely biopsy, that's autopsy, surgery, and so forth. It's been putting brain cells in vitro and trying to understand them isolated from this enormous network. And of course, from 2 points of view, I think the Human Genome Project has taught us enormous lessons about big science and what it can do. So I'll start by defining exactly how I think the Human Brain Project is different from the Human Genome Project. So for the genome, it was really well defined. We were principally going to get the sequence of the human genome. The Brain Project is infinitely broad. In fact, we don't even know probably some of the most important questions we should be asking. The Genome Project's resources were much larger, were very well defined, were over an extended period of time, and as you heard from Jim earlier, we're not even sure about the 100 million that's been proposed for the Human Brain Project. And finally, I would say a really fundamental difference is the Human Genome Project incubated for 5 years, starting in 85 through 90, with an enormous amount of ferment in many different kinds of communities. And initially, the reaction was enormously negative. Maybe 90% of the biologists were against the Human Genome Project. And gradually, it swung around. Ironically, the people who really favored it from the very first were the politicians. And they were attracted to the idea of world leadership in an important area, of maybe it will do something for medicine, but most important, they were excited by economic opportunities. So what exactly are the benefits then of the Human Genome Project? And I would say for the benefits that I'll list quickly, I think they could be extended in analogous ways to the Human Brain Project, and we'll see if you agree. One thing the Human Genome Project did was to give us the complete parts list of human genes and by inference proteins, and that opened up the field of systems biology. The parts list was really key to systems biology. Another thing it did was to democratize every region of the gene. That meant every region was available to every biologist to whatever extent they wanted. A third thing it did was really revolutionize and pioneer new technologies, both for doing genomics, but it moved to proteomics, and then into the computational and mathematical area. And what that did was irreversibly brought to biology this cross-disciplinary cadre of scientists engineers, mathematicians, and so forth. And that has irreversibly changed biology in very, very good ways. It enabled mass spec-based proteomics. You needed to know the sequence of the peptides before you could determine their mass. It pushed the open source for software, and the idea that the tools would be democratized and readily made available was one critically important aspect of it. The second important aspect was immediate publication of all data. And that made the data available to the community for analyses. And I think these are two fundamental principles that have been really revolutionary. It gave us access to all the genomes of all creatures, and in doing so, it's transformed the life sciences, and it's absolutely revolutionized our understanding of evolution. It's demonstrated that all organisms do have that single initial origin, and this has been established in many, many different ways. It's begun to transform medicine by creating systems medicine. I'll talk a little bit about that in my talk later on. Personalized medicine and systems approaches to diagnostics and therapeutics. And as Jim said, it's opened up this whole field of pharmacogenomics. That is the idea that different humans react differently to drugs. And that's a really important thing to know in many cases. It changed the sociology of biology. It spent a fraction of its money on ethics and privacy and the legalities that had to be worried about, and many, many other things. And of course, it raised this whole issue of big and small science. The genome is an example of wonderful big science that contributed enormously, and yet in these days of difficult budgets, the NIH reverts again and again to let's get rid of all big science and only fund small science. That would be the death toll for our leadership in the world of science. You need a mixed portfolio of big and small science. They are enormously complementary to one another. Battelle made this incredible estimate about 2 years ago that for an investment of $3.5 billion, the genome has returned $800 billion. And if that's true, that's a pretty incredible return on investment. So these are all things that I think open up possibilities for the BRAIN project as well. So how does systems biology and systems medicine deal with the biological complexity? And of course, the complexity of the brain is probably unparalleled by any of the other human biological systems. There are really these five fundamental approaches. The idea biology is an informational science that organizes our thinking. The idea that you need to create appropriate infrastructure to be able to do big science in a directive, integrative, systems-driven manner. The essence of systems biology is to capture the dynamics of what's happening in living organisms, and then to develop technologies and strategies that will open up new windows of opportunity while at the same time creating the appropriate kind of analytic tools. So what about this idea of biology as an informational science? Well, there are two fundamental types of information: your genome and the environment to which that genome is exposed. What is interesting is those two types of information generate your phenotype. Through development, through physiologic responses, through disease, through the aging process. And of course, what connects these two types of information with phenotype are dynamic biological networks and molecular machines that are themselves dynamical creatures. And those are the focus of much of systems biology at a current time. There are the two types of information generate a whole hierarchy, in a sense, of subtypes of information, and these go from DNA and RNA and proteins and interactions and networks and cells and organs all the way up to ecologies. And the really important point is the environment impinges upon each and every one of those subtypes of information and modifies the core digital output. So if you're to really understand a system, you have to be able to integrate these different types of information in such a way that you make explicit the environmental contributions, and that's one of the grand challenges of systems biology. One way that you can deal with all of that information is to create for each individual this so-called network of networks. You have genetic networks and molecular networks and cellular networks and organ networks and networks of individuals, and that network of networks in disease becomes perturbed, and the modification of that network of networks explains both pathophysiology of the disease, but if you can delineate it, gives us fundamental new insights into diagnostic and therapeutic, and ultimately into preventive approaches. And it's a great way to organize complex sets of information. And this is a major area at ISB that we're really pushing at this point in time. The mantra that we started ISB with back in the old days was leading edge biology always has to push the development of new technologies, and they in turn mandate the need for new analytic tools, computational and mathematical. And if you succeed in doing that, then you can really revolutionize biology. To be able to do that, you have to create this cross-disciplinary environment with all of the types of scientists seen at your right-hand side there. And what they must do is, one, learn to speak the languages of one another, and two, learn to work together effectively in teams. And if you can do that, you can not only generate biological information, but this whole mantra of biology driving technology, driving computation at each turn creates new concepts, new technologies, new types of software. And those further science, but they open the possibility for innovative creation of new companies. And ISB, I think, has been particularly effective in taking advantage of those opportunities. What about the dynamics of living biological systems? How do we go about studying them? It's really hard to study in humans because they can't be manipulated. So one of the ways we've started studying them is looking at animal models, which we think are incredibly important. And for a given system, what you want to be able to do is, one, enumerate all the parts that are present in that system, and two, then extract the nature of the dynamics of these networks and so forth, and then three, convert this into models that are both predictive and, in the case of medicine, can actually be actionable. And how you integrate these different endeavors together is the essence of systems biology. And I think really to be doing systems biology, you have to be able to effectively integrate the kinds of needs that we've talked about here. So I would say there are really two fundamental essentials for systems biology. One is the idea that you look at the system you're interested in, you formulate models from the data about how it works, you create hypotheses to test them, and then you perturb the system to test those hypotheses in an iterative manner, so you bring the data you have into accordance with the model that you have in mind. And ultimately, what you need to be able to do is create models that are predictive and actionable, and they need to be able to explain the emergent behaviors of the system. I think the second essence of systems biology is how we think about data. It should be global as possible. You ought to measure all the components, and we can do that for genomics, but not for many of the other omics. You want it to be dynamic, not just temporal, but spatially dynamic as well. You need to integrate different types of data. And finally, you will be generating large data sets. There are enormous amounts of noise in these data sets. And the essence of systems biology is it gives you a cadre of very powerful tools for extracting signal from an enormous amount of noise. Technologies here at ISB that we're pushing for the future include third-generation sequencing using nanopores, nanochannels. And this is not us per se, but a company that lives here that's doing this. And I think the exciting thing about the company that succeeds in doing this is it's going to make the genome inexpensive. It's going to give us very long reads, so we can do de novo assembly of human genomes. And it'll do at the same time epigenetic modifications on the DNA at very reasonable error rates. A second area that ISB has always been pioneering in is proteomics. So we pioneered with Rudy Ebersol and Rob Moritz targeted proteomics, and we're beginning to do the same for an area called swath proteomics that gives us the potential for making proteomics global in exactly the same way large-scale sequencing and DNA arrays made genomics global. And I think this is really going to be exciting. The idea of peptide protein capture agents, you'll probably hear from Jim Heath. I think it's critical we find a substitute for antibodies, and there's a great one that is coming down the line. The maps that we look at in terms of networks are both incomplete and horribly inaccurate. We need better tools for creating these network maps. And again, we're using proteomic and microfluidic techniques to be able to do that. Single cell analyses are going to be important. In a sense, the single cell analyses does for biology what systems medicine is doing for medicine. It allows you to focus on the individual person, the individual cell, and study them in the context of what are their quantized individual properties, and then aggregate them into groups that share related kinds of properties. And the idea of quantized cell states, I think, is really going to be an important conceptual state for the future. And finally, we're using a nanostring instrument, a spin-out from ISB, to analyze not only single molecules of RNA and microRNA, but single cells and antigens and proteins as well. So it's going to be very powerful. The strategies that we've developed here, family genome sequencing, systems-driven blood diagnostics, I'll talk about both of those this afternoon. I won't talk about IPS differentiation, but it's going to be a really key approach both to understanding disease mechanism and the stratification of disease. The analytic tools, one of the strengths of ISB, big data and storage and analysis. How do you capture and validate and store and mine and integrate and ultimately create predictive models? And I think multiscale modeling that's really being pioneered by Ilya Smilovich is really going to be critical in the future. And I'll close with a new kind of partnership that ISB is beginning to create now, and that is systems-driven clinical partnerships to attack big clinical problems. So we're studying with Nova 750 families, trios, husband, wife, infant, to try and determine the causative diagnostic factors that will give us insight into preterm birth and how to control it. This costs the US and, in fact, developed and undeveloped countries enormous billions of dollars per year, and it's an ideal area to target. We've already got 2,200 genomes that we've looked at, and there are some really interesting and revelatory data in this particular regard. And more recently, we're putting together a study where we'll look at 7,000 patients with regard to atherosclerosis with very good phenotyping by imaging, CT scanning, together with detailed molecular analysis of the blood and the white cells in the blood, and the complete genome sequences of all 7,000 of these patients. And it'll be an ideal model in which to practice the idea of data integration. It'll be done longitudinally, and part of it will be done in families for reasons that you see as being very important. So to close, I'll just return again to the theme of complexity. And my view is that all the disciplines, scientific and engineering, struggle with the challenge of complexity in the 21st century. And I think what's unique about biology, it has these ways to approach complexity that we've discussed in the course of this talk. And of course, what that opens up the possibility is looking at the big problems of society beyond healthcare and the brain and things like that, to animal health and nutrition and wellness and the environment and energy and agriculture. And the idea is, with this approach to complexity and the kind of platforms and culture we've generated at ISB for taking on big science problems, I think we have unique opportunities to attack in very big ways and bold ways many of the deepest problems of society. And of course, the real issue will end up being funding. So let me just say, so I'm finished, but let me just say we're going to hold questions until the panel discussions at the end. All the speakers will be members of the panel, just so we can go through efficiently. So I'd like to turn to an introduction of Christoph Koch now. And actually, everything you need to know is in the book. There aren't, I mean, it's, I think, very well discussed there. But I would say a couple of things about him. I was at Caltech as chair of the Department of Biology in 1986 when Christoph came, I think, from MIT at that time. And we were really excited to get him. I mean, one of the things that he was excited about was this area that I've always thought was overwhelming of consciousness. And he struck up a little bit later a collaboration with Francis Crick that I think was absolutely a classic pioneering attack and approach to this book. And in fact, what about 6 years ago or so, you published a book on consciousness, which really is a marvelous thing to read. You ought to look at it. So anyway, he's now at the Allen Institute as Chief Scientific Officer, and he's interested in a large-scale project that we're going to hear all about. So Christoph, please.